Uh, Thank you very much for the introduction. I'm assuming that you guys can hear me okay?
Yeah, it's good.
Okay, great. Um, Great. So I am really excited to kind of share some of the work that's been going on in my lab here with you guys, and really want to thank the organizers for inviting me and giving this opportunity to speak here. And so I'm going to tell you about work that we've been doing in terms of developing methods to really study genome organization and DNA methylation at a single cell level, and how we're applying this in the context of sort of normal cell types in development, as well as some ongoing work about how we are trying to apply these kind of methods in the context of cancer genomes to really understand aspects of cancer genome evolution. Um, Okay, so I always kind of start my talk. I'm interested in this question of how our genomes are organized in three-dimensional space inside of cells. And I always kind of start by saying, we're used to thinking about genomes as these one-dimensional sequences of nucleic acids that we can, of course, represent here as an actual sequence of bases, but maybe more commonly we will represent in sort of a map here where we can annotate particular features of the genome. And this could be things like genes and different regulatory elements. Um, but ultimately, we know that our genomes are actually folded in three-dimensional space inside of cells, and how our genomes are folded actually has a major impact in terms of how a variety of different processes in the nucleus work. And this includes things like DNA replication, DNA repair, as well as gene expression. And so this question of gene expression and how this relates to genome organization is really why I ever got interested in this kind of question or problem in the first place. And this is a very typical problem that if you're interested in regulatory elements, you're using genome-wide tools for mapping these, you'll inevitably come to situations where you identify something like an enhancer or other regulatory element in the genome. And from the one-dimensional sequence alone, it may not necessarily be obvious sort of what is the target of this regulatory element in the genome. And so one of the major reasons why we're interested in this problem of how our chromosomes fold up in space is that the dominant models for how enhancers work is that they're brought in close spatial proximity to the genes that they regulate. And this causes proteins that bind at the enhancer to be able to interact with proteins that bind at the promoter, and this can facilitate transcription. And so we think that this is a really interesting question for a variety of different ways that, by understanding how our genomes are organized, we might be able to build maps of which regulatory elements target which genes in the genome, as well as trying to understand what are some of the molecular players that actually might drive these kind of physical associations over these wider genomic distances. And so we're also really interested in trying to understand, given that enhancers are known to be features that actually are critical for lineage specification, we're also really interested in understanding how genome organization actually varies across cell types and how this may contribute to cell type specific communication between regulatory elements and genes. And this is actually an example from data that we have in a recent preprint where we're doing this in iPS cells. But this is again using kind of bulk approaches where we're differentiating these cells and being able to do these kind of assays using sort of conventional bulk methods. And it would be really nice if we could apply these same kind of tools, these Hi-C assays, for mapping how our genomes are organized in the same way that I think that people currently are applying tools like single-cell RNA-seq, where you can apply these complex systems and then really in silico use the data to actually reconstruct the different cell types within a particular biological system or tumor or tissue. And then from that actually learn things about the kind of cell type specific patterns of expression and how they vary. And so this is something we've been interested in for quite a long time, but there's actually some technical challenges in this. And I could highlight a couple of these here in terms of why this is really hard to do for trying to map out how our genomes are organized in space using single cell tools. And so one of these is a problem that's called intrinsic heterogeneity. And this is shown here. This is a slide from a paper from Tom Mastelli's lab, where they're doing fish and they label individual genomic loci with these probes. And these are all in mouse embryonic stem cells. And so what you actually see here is, say, in a nucleus over here, the probes are all really spread out across the nucleus, whereas in the cell over here, they're actually all at one pole. There are some cells where the probes are all on top of each other. There's some where the blue and the red are on top of each other. There's some where the red and the yellow are on top of each other. And these are otherwise supposed to be the same cell type. And so really what this says is that there is not a deterministic feature that organizes our genomes within individual cells, right? You don't always have the exact same chromosome position in the exact same place from one cell type to the next. There's a level of randomness to it. And effectively, that randomness amounts to noise if you're trying to use this as a single cell assay to begin to identify cell types. The other kind of major challenge that we also know contributes to this is that there are dynamic processes that occur in the genome that will actually actively reorganize our genome. And so a great example of this are processes like loop extrusion, which is this sort of molecular process driven by cohesin that essentially gives rise to these kind of chromatin features that we know like TADs or chromatin loops. And again, this dynamism, it's again within a cell type where the identity may be fixed. These kind of processes are taking place and essentially introducing variability between individual cells. And the result of this is that the earliest work really developing single cell Hi-C type methods and using methods to embed the data in low dimensionality space, really the dominant features that emerge are that of the cell cycle. And on some level, that's fine. We know that chromosomes are actually massively reorganized when the cell goes through cell division. But if you're interested in applying these kind of methods to like a tissue, this is not necessarily the biology that you want to be able to extract. And so we reasoned that this was something that we really needed better tools to kind of address these kind of problems. And so one of the things that we have focused on, and I will also say this is the work that has been initially started in my lab by Dongsoon Lee, who's a former postdoc in the group, is really trying to develop methods to look at more than one thing at the same time. And so in this case, what we're talking about is looking at both DNA methylation and 3D genome structure simultaneously in single cells. I also say a lot of this work has been done in collaboration with Joe Ecker's lab, and this is really sort of an ongoing collaboration that we've had now for several years. So these assays that we developed that we call single nucleus methyl 3C sequencing, or SNM3C-seq, the way that we perform these experiments is we start with a conventional experiment that is like a Hi-C or a 3C experiment. And in a Hi-C or 3C experiment, we take a population of cells, we fix them with formaldehyde. This preserves protein-protein and DNA-protein interactions. And then we digest them with a restriction enzyme of interest. And then you ligate these free ends of DNA together such that regions that were in close spatial proximity can now become covalently linked to each other. Now, this is actually all happening within an intact nucleus. And what that allows us to do then is to sort nuclei into wells of a 384 well plate. And then we can subject this to bisulfite conversion. So what bisulfite does is it will take cytosines, unmethylated cytosines, and it actually causes a spontaneous deamination that converts them into uracil. And methylated cytosines remain unaffected by bisulfite. And so what ends up happening is this allows you to read out the methylation status of individual cytosines in the genome and distinguishing bases that were either methylated or unmethylated because the uracil is read out as a T in sequencing. And at the same time, you get the presence of these chimeric reads that are actually derived from these ligation events between spatially proximal features. And so you can actually read both of these out simultaneously from the sequencing run, and we go through a single cell library prep to basically generate this kind of data. So we published this method first a few years ago now, and we've been applying this in a variety of different contexts, but one that I'm particularly excited about, and that I'm going to tell you about some of our work today, is actually applying this really across a wide variety of human tissues to really try to generate an atlas of both 3D genome organization and DNA methylation across human tissues. So we've done this now for 16 different human tissues. These are from otherwise normal, healthy postmortem donors. Actually, most of these tissues were profiled as part of the NTEX project, which is this project between GTEx and ENCODE. So there's actually a lot of additional omics level profiling of these, both at a bulk level and at a single cell level. In particular, there's a single cell attack-seq data for essentially all of these lineages done by Bing Ren's lab. So we have this data, we have the single cell population from each of these tissues, and then we can use this to try to identify the constituent cell types. And there's a variety of different ways that we try to do this, and I'll tell you about some of the different efforts here. But for the most part, when we use this clustering, we're using sort of a joint clustering approach where we consider information from both DNA methylation as well as 3D genome organization. And in total, we identify what we're calling sort of 35 major cell types, and then 206 cell subtypes upon sort of iterative or secondary clustering of these primary and major cell types. So there's obviously a lot of data here, but I want to kind of touch on a few interesting aspects of this that I think are particularly cool findings from the study. The main paper is actually on bioRxiv if you want to read it. And so there's kind of a couple different features that I want to explore in terms of some of the analysis that we've been able to do. So one of them is looking at features like chromatin loops. A second is looking at genome organization feature called chromatin compartments and how we think this is related to features that are called partially methylated domains in the genome. And lastly, the very kind of interesting case is we can find instances where the DNA methylation patterns and the genome 3D genome structure patterns actually disagree within potential tissues and lineages. And we actually think they disagree in ways that are telling us something about the biology that we think is really cool. And I'll sort of highlight this at the end. Okay, so in terms of this aspect regarding chromatin loops, so these are these kind of features that I mentioned at the beginning that sort of bridge sort of distal regions of the genome together. And we can basically ask where and when do we find this across all of these different major cell type classifications. And one of the ways we do this is for each individual cell, we basically calculate the frequency of chromatin interactions as a function of distance and plot this here as a heat map. And we see there's kind of two major peaks of signal given relative to what we would expect by chance. One of them is here around 500 kilobases in size. And this corresponds to features like loops and topologically associated domains in the genome. There's a second peak that we can find here that is sort of centered around maybe 50 million base pairs. And this corresponds to these much larger chromatin compartment type patterns that are related to things like A and B compartments or sort of heterochromatic and euchromatic organization in the genome. And so what I think is interesting is that we actually can see these features across all the cell types, but they seem to be used differently depending upon lineage. So there are some cell types like these different classes of excitatory and inhibitory neurons that have really strong signal in this range related to sort of TADs and chromatin loops, but actually have a quite weak signal related to these larger scale chromatin compartment features. Likewise, you can see at the other end of the spectrum, there are cells, and this is actually largely in hematopoietic lineages, that have relatively weak signals at the level of these kind of TAD and loop type regions of the genome, but seem to primarily be using this kind of chromatin compartmentalization pattern as an organizing principle for their genome. We can see this both at this kind of global contact level, as well as if we go in and do things like call individual loops and ask how long are the loops or how many loops you have. And these kind of patterns seem to actually hold depending upon lineage. And so what we think this means is that there's actually differences in which ways cells use these different ways of organizing their genome depending upon cell type and cell lineage, which we think is particularly interesting. And we do think that there are some patterns here. So, neurons seem to have much more of these domain-rich organizations, whereas a lot of hematopoietic lineages seem to have much more of these compartment-rich type organizations. So in addition to these kind of like global classification schemes that I'm talking about here, we can also go in and identify like loops in individual cells. And this is kind of a case where we can see this here. We can see that this is a loop that we find. This is in 3 breast epithelial cell types. This is the luminal hormone sensitive, luminal secretory precursor, and basal myoepithelial cells. And so you can see this loop here forming in the basal myoepithelial cells. It's really absent in these other lineages. And this is nice because this is actually at the oxytocin receptor gene. This is a known marker gene of this lineage. And so we think that this is likely an indication of sort of some of the mechanisms of how this is actually being regulated in this cell type specific way within the basal myoepithelial cells. And so we can actually go through and test this a little bit more explicitly. We can actually look for genes that show differential expression between these related lineages. And we can indeed see that they're associated with patterns of differential chromatin looping between cell types in ways where when you have this kind of chromatin looping present in a particular lineage, it's associated with expression of those genes. And so we can expand this analysis of these differential chromatin loops really to kind of a genome-wide or body, tissue-wide scale, and really beginning to then cluster loops according to both where these are in the genome as well as what lineages these appear in. We can actually see that there's some kind of interesting patterns that emerge. So these are essentially loops that are all sort of more frequently found in these different fibroblast lineages. These would be ones that are found more frequently in neuronal lineages and some of these epithelial cells, as well as hematopoietic lineages. And so what I think is kind of interesting about this is we actually see this kind of shared structure of where we find these chromatin loops across different classes of cell types that I think are really being organized along these kind of almost histological classes of cells, like so fibroblasts or epithelial or mesenchymal lineages. And I think one of the cool things that this also allows us to do is to go in and ask questions about what actually controls this kind of cell type specific looping events. So CTCF is basically well known as kind of the major player along with cohesin that actually demarcates where you form these kind of chromatin loops in the genome. And we can see that the CTCF motif is enriched essentially across every single different cell type in terms of where we find these kind of cell type specific chromatin loops, which is nice. That suggests that these are forming through the mechanisms that we think that they should form through, but it doesn't actually tell us a lot about why they're cell type specific because CTCF is really expressed like a housekeeping gene. But I think looking at these kind of secondary hits, you can actually begin to see that these are indeed much more lineage specific. And I think what this suggests is that these sort of secondary factors are actually kind of critical for likely providing the signals that help open up chromatin and allow CTCF to bind potentially in these cell type specific ways to really give rise to these kind of patterns of cell type specific looping that we see. So the second part, that was the first part I was going to tell you about in terms of chromatin loops. The second I'm going to tell you is about some interesting things that we can see related to chromatin compartments in these regions of the genome that are called partially methylated domains. And so the way that we kind of come across this part is we're starting to just look at global patterns of DNA methylation across the different cell types. We can see that there's actually a pretty wide variety here. So some cells have around 80% methylation, others it's less, around 60%. And we can actually see that if at an individual level, the base composition tends to exist in this really bimodal distribution. So most cytosines are either fully methylated or fully unmethylated. But you actually get some cell types where you get an enrichment of these partially methylated cytosines. So basically things that are not either fully methylated or fully unmethylated, but exist in this kind of in-between state, maybe 50% methylation or so. And if we go in and examine sort of genome browser tracks, we can see that these are actually clustered quite frequently in the genome. So you can see these sort of long stretches of the genome that show these partial methylation phenotypes adjacent to other regions of the genome where you see this more enriched or more sort of full methylation patterns. And this has actually been well known. This has been described many times before, and these are called partially methylated domains. There are these long segments of our genome that are associated with these kind of reduced hypomethylation states. And what we think is actually kind of interesting is these are actually very well described in the context of some cultured cell lines as well as cancer cells. But in this atlas, we can now see evidence of these occurring in specific lineages. And so we are actually really interested in characterizing really where these kind of partially methylated domains show up, what cell types are affected by them, and what region of the genome. But this is actually kind of challenging. So when we use existing tools to try to find where these partially methylated domains were, we can find regions of the genome where these methods actually work quite well. They can identify these regions of hypomethylation. But in other lineages, these tools didn't actually work as well, and it became kind of hard to explain or understand like why these were having these lineage-specific effects. And so one of the things we did is we kind of took a step back and we tried to characterize the data in a very simple way, which is essentially just taking our genome, breaking it up into 10 kb bins, and then asking about the distribution of methylation levels across individual cytosines in each 10 kb bin in the genome. And this is basically what we see. So if we take the whole genome, we cluster it into 10 kb bins, and we cluster by these distributions, we actually get 4 clusters that emerge. There's 2 very small ones that are almost impossible to see here at the top that are either fully unmethylated or a really bimodal. So it's a mixture of fully methylated and fully unmethylated cytosines. But the bulk of the genome really consists of one of 2 states otherwise, which is either a fully methylated group of regions of the genome, or a set that are existing in this much more heterogeneous state where we see this enrichment of these kind of partial methylated phenotypes. And so what was nice is we could go and actually then look in other lineages and see that these same kind of approaches worked. But the kind of interesting thing that we could see is when we compare related cell types, we can see these cases where, so this is in T memory cells, we see this large hypomethylated block. If we look at naive T cells, so related lineage, we see that the same region of the genome shows this relative hypomethylation. But if you notice the axis change, so the degree of hypomethylation here is much more strong in these memory T cells than in naive T cells. And indeed, we can apply these same kind of clustering approaches even in cell types that don't have really obvious partially methylated domains. We can see the evidence of these same kind of clusters emerging, this sort of fully methylated cluster, this more heterogeneous methylated cluster, and then these sort of very bimodal or unmethylated regions of the genome. And what's really interesting is if we then ask, okay, in memory T cells, what does this look like? You actually see the same patterns, but you see a greater loss of methylation in this kind of partially methylated or more heterogeneous compartment in the genome. And so one of the things that we think that this means is that probably all cell types have some level of partially methylated domains. And it's just really a degree of the extent to which these regions have become demethylated over time. And so the other thing that we think is really interesting about this is we can actually see that this shows a very clear relationship with these kind of A and B compartment patterns of our genome that are related to spatial genome organization. And so what are these A and B compartments? So these are this kind of very large scale organization of our genome. This is identified over 100 years ago through electron microscopy, where you have this partitioning, again, a spatial partitioning of our genome into either euchromatic or heterochromatic regions, but again, they're spatially distinct from each other. And so this kind of pattern actually emerges really clearly in Hi-C data, where you can see this kind of like almost plaid or checkerboard-like pattern, and with some sort of relatively simple mathematical transformations, you can kind of enhance this. And what this really shows is that these euchromatic regions interact with euchromatic regions, heterochromatic regions interact with heterochromatic regions, but they don't talk so much to each other. So we can apply these same kind of transformations to our data, identify these kind of chromatin compartment patterns, and then really begin to compare this with regions of the genome in terms of methylation levels and methylation density. And so what we can see is these regions that we're labeling here in blue are these more heterochromatic sites in the genome. They're very clearly correlated with the presence of these partially methylated domains in the genome. And not only do we see this at a global level, we can actually see evidence of cases where this switches. So this would be a region of our genome here that's an A compartment in these fibroblast regions, where they're active compartment, but that switches to this inactive B compartment in other lineages. And you can actually see that the kind of methylation phenotype also follows the same pattern. So this is in this more fully methylated state in the fibroblast lineages and becomes this more partially methylated state in other cell types. We can show that here also just in kind of example browser shots where we see these kind of peaks in our A and B compartment score, suggesting this is an active A compartment region that correspond with higher levels of methylation, and these dips in these kind of partially methylated phenotypes correspond to these B compartment regions. And so one of the things that we think is cool and interesting about this is I think this potentially suggests that the way in which we regulate or reproduce methylation during cell division is actually really going to be spatially isolated and spatially distinct. And so we think this is actually really interesting that we've been able to identify these kind of unique organizations. And we think this is actually occurring really regardless of lineage. It's just the degree of hypomethylation in a partially methylated domain can vary depending upon cell type. So that was the second part. And so the last thing I was going to tell you in terms of our analysis of these kind of normal tissue atlas level data is actually about some really interesting cases where we see discordance in the patterns of DNA methylation and genome organization across cell types. And so how we kind of get into this kind of question is as I mentioned at the beginning, we have two different data types here. We have DNA methylation and 3D genome organization. And we can use both of them to kind of jointly cluster the data. We can also use each of these to cluster the two lineages or classes of cell types independently. And so if we do this at this major cell type level, we see that both of these methods identify most of these 35 major cell types equally well. But what's interesting is that if we then use each method independently to do this kind of subclassification, we can actually see that there's cases where there's differences, where one method identifies one set of clusters and another identifies a different set of clusters. And so we can quantify this here using an adjusted RAND index, where we're essentially asking how well do the two clusters agree upon this sort of secondary iterative clustering between DNA methylation and genome organization. And so there's some lineages where this actually agrees fairly well. This is particularly the case in these kind of neural cell types. But there's actually a reasonably large number where there's actually relatively little agreement in terms of these subclassifications that we observe using these two data modalities. And when we kind of dig into the cases, I think it's super interesting. And so this is an instance where we see different clusters that emerge in the context of skeletal muscle. And so this is showing if we basically take skeletal muscle cells, we cluster with the chromatin contact or 3D genome data, we get three very nice distinct clusters. These correspond to a satellite or stem cell population, as well as fast twitch and slow twitch fibers. If we do the same thing with DNA methylation, you see this partitioning of the fast twitch and slow twitch fibers, but the stem cell population is actually not clearly as distinct. In fact, it almost looks like there's like a trajectory here going from the stem cell population into each of these more differentiated lineages. And this actually becomes much more apparent when we show the levels of DNA methylation across individual cells here, which is that the DNA methylation-based clustering is really actually picking up on these gradients of DNA methylation levels, such that the sort of stem cell state is associated with this relative hypomethylation, global hypomethylation state, and that you gain global levels of methylation upon differentiation or maturation into these mature muscle fibers. And so what we think is really cool about this is that this is much more distinct in the context of 3D genome structure. So the genome organization may be telling us more about cell state classification. So is this a stem cell? Is this a fast twitch fiber? Is this a slow twitch fiber? Whereas the DNA methylation patterns here are actually telling us something about differentiation state or maturation. So it's that we could have a cell that looks like it has relatively low levels of methylation, which may be a recently differentiated myofiber. And so we think this is actually really nice because you can use this maybe to learn sort of additional pieces of information about this system by examining these cases where these two things actually don't agree with each other. The other thing that we can see is we can see cases that are even almost more extreme. So this is a case where we're looking at epithelial cells of the stomach. If we try to use these for clustering based on DNA methylation, we actually don't really pick up on particularly clear patterns at all. Whereas from the chromatin contact data, we actually identify really distinct clusters corresponding to kind of known cell types within the sort of epithelium in the stomach. And so this is kind of interesting, but one of the kind of cool parts about this is if you take the cell type labels from the chromatin contact data and you use that to call differentially methylated regions based on the DNA methylation patterns, you can identify regions that show differential methylation between these different epithelial cell types, and you can actually see that they're correlated with or associated with genes that are showing cell type specific expression in a correlated way. And so what this means is it doesn't mean that the DNA methylation is actually devoid of information here. But what we think it means is that the cell type to cell type level variation in DNA methylation in this particular context is actually less than the variation in DNA methylation you see between any two individual cells. And we actually don't really understand what's the basis for this. We actually think this is really interesting, like why some lineages show this really strong agreement between DNA methylation and 3D genome organization, whereas others show sort of less agreement, either in these cases where there's relatively little information or greater variability, or these cases where there might be instances of sort of degrees of differentiation. And it's not totally clear, but I have a speculation that some of the cells that agree the most between these two modalities are actually fairly long-lived cell types. So we see these in neuronal lineages in particular. And so what I think this might mean is that there's actually a level in which these two features may mature over time as a cell differentiates or divides, such that they only over maybe significant periods of time do they actually really retain the exact same sort of cell state classification information. So this was sort of the first part of my talk, and I'm just going to kind of summarize here. So we've been using these kind of single cell tools that we've developed to really build these atlases of 3D genome organization and DNA methylation across human cells. We're really interested in using this to try to understand things like the patterns of chromatin loops and chromatin compartments and how this relates to underlying differences in DNA methylation. And lastly, we think that there are these actually really cool and really interesting cases where you can see this divergence between DNA methylation and genome organization that we think suggests that there's ways in which we can use this to really learn different biology of the systems. And so this work is actually all in bioRxiv. And so I'm going to, in the last few minutes here, move on and tell you a little bit about ongoing work in the lab where we're trying to take these same methods and apply these in the context of cancer genomes. And so one of the things that our lab has been focused on in the past is actually trying to understand how structural rearrangements of our genome, things like translocations, inversions, duplications, may rewire our higher order three-dimensional genome structure. And so, some of this stems from basically doing high-C and different data sets where this is basically an example of normal chromosomes. So this is a normal chromosome 1 on the left, and then on the right is the interaction between chromosome 1 and chromosome 2. You see that on chromosome 1, there's this very strong signal along the diagonal that's related to the fact that the chromosome is a polymer. And there's a relatively weak or sort of lack of signal between the two chromosomes. But this really differs dramatically when we start to look at rearranged genomes. And so this is actually looking at K562 cells where there's a variety of different rearrangements. And in particular, this is looking at one specific case where this is an interaction between chromosome 10 on the x-axis and chromosome 3 on the y-axis. And this is the site of a known translocation in these cells. And at the region where there is this translocation, there's this very, very sharp demarcation in this kind of interaction frequency signal. And this is basically the signature of a translocation in a high-C data set. And so we've actually developed tools in the past to essentially use high-C data here to directly identify where these kind of structural variant signatures take place, and have actually used this as a tool to try to identify structural variants in cancer genomes. And comparing this with things like bionanocal optical mapping and whole genome sequencing, we can actually see that high-C is actually quite sensitive, almost on par with things like whole genome sequencing for identifying what we're calling sort of large-scale rearrangements. So these are sort of large-scale intrachromosomal events or interchromosomal rearrangements. And we think that this is actually really nice because the signal in the high-C data can actually expand over megabases from the breakpoint, and it can actually be detected in cells that you have relatively low coverage. And so we think that this is actually a cool way to really think about applying this actually in the context of single cells for single cell structural variant analysis. And so we've been doing this now in the context of some primary patient samples, and I can show you some of the data and some of the things that we think we can learn from this, which I think is kind of cool. So this is from a glioma patient. This is looking at high-C data across the entire genome. So this is chromosome 1 in the top left, all the way down to chromosome X in the bottom right. And you see most of the signal is within chromosomes, but there are these regions, these kind of strong enrichment signals that you see going between different chromosomes that is really corresponding to the signature of these structural variant patterns. And so what we've been able to do in these data sets is we can use these tools, this tool that we call high-C break finder, to identify structural variants. And this is actually showing an exact case where this is running and identifying a balanced translocation. And these little black boxes are actually the region that is being identified and called by the tool as the sort of site where the breakpoint is, or the breakpoint proximal region. So we can identify these regions of the genome that are basically the signatures of structural variants, and then we can actually use that specific signal to cluster our cells in low-dimensional space. So we're not looking at genome-wide signal, we're not looking at anything related to cell type or lineage or identity, we're really just looking at structural variant pattern here. And so we can do this, and we can actually embed the cells in low-dimensional space and really see these kind of patterns emerge that we think are related to sort of cell type identity and potentially tumor evolution. And so in this case, you find sort of a handful of different clusters, and at least this one on the right is very clearly sort of normal cells. And we can see this because if we then regroup these single cells, we regenerate these kind of bulk contact maps, we can see that this cluster on the right is sort of the normal cell population. You actually lose a lot of these long-range kind of interchromosomal rearrangement patterns. There's actually some kind of faint signatures of copy number changes that are also gone on the right when we look in the normal cells. And so at the same time, you can actually see that there's sort of multiple tumor clusters that we call on the left-hand side. And what we think is actually really interesting about this is this seems to be a case where we can identify an instance of an ecDNA that's actually undergoing some level of almost clonal evolution here. So we can actually identify two different tumor cell clusters, and then the sort of string of cells here on the top that is highly related to the copy number of a single locus in the genome is this single locus on chromosome 11. And this appears to, basically on the signature here, this actually appears to be like an ecDNA type event. And so we think this is really nice because we can actually use this to then begin to understand sort of what is the differences in how these kind of genomic alterations, in this case in ecDNA, may be altering the organization of our genome, the methylation landscape, other sort of regulatory features, as well as potentially learning things about how this arose and aspects of tumor evolution. Okay, with that, I'm going to stop and I can sort of acknowledge some of the people who did the work. I mentioned earlier that a lot of this work started in my lab by Dongsun Lee, who was a postdoc in my lab at the time. He's now at Seoul National University. The current work that's being done in my lab that's really related to this project is being done by Tessa Pope, who's a postdoc in the lab, Yunwen Wu, who's really leading a lot of the development of sort of next generation versions of these single cell assays that we're working on. Ami Pant, who's also working on these kind of approaches, as well as Tula Keel, who's leading a lot of the glioma-based work in our lab. And this has really been an ongoing collaboration for quite a while with Joe Ecker's lab. And a lot of the work that I was showing at the beginning, in particular the sort of body map type data, was led by Jingtan Zhou, who is now in a kind of independent fellow position at the ARC Institute in the Bay Area. And with that, I will stop and take any questions.
Thank you very much. Okay, there is one question already on the web. The question is in your SLM3C data set across different tissues, were there any particular tissues where the clustering was less distinct or more challenging? For example, lung tissue like that.
Yeah, so there definitely are some cases where I think it's more challenging, and I think this is in particular the case for thinking about some of the methylation patterns like I showed in the stomach. So the epithelial gastric cells are one of the more challenging ones. There's actually some of the cells in the colon are also a little bit more challenging, at least from a DNA methylation standpoint. It's not entirely clear why that's the case. I think that there's a level that, some of these cells are turning over. And so if there is a level where the methylation patterns sort of need time to mature or harden to reflect sort of these native cell states, I think that may be related to aspects of cell turnover, but it's not entirely clear. The other one that I would say that is absolutely very hard is placenta tissue. Again, from a standpoint of DNA methylation, placenta has these really strong patterns of these partially methylated domains, and they're actually really variable between individuals. And so at a first pass, most of the clusters that we get from the placenta are actually just individual specific events. And in some cases, it's not even clear that they're different cell types. They may just be almost different subclones within in the placenta in terms of DNA methylation levels.
Okay, the next question is this. It's natural that 3D contact information changes with the cell cycle, but have you ever compared 3D contacts across different cell types within the same cell cycle state? For example, were the comparison made among cells in the same phase, such as S phase?
Yeah, so for our data set here, for technical purposes, we actually are entirely sorting out cells that are not in G1. The main reason for that, it has to do with doublet identification. So we have to fix the cells for these high-C assays. We can see fairly clearly that leads to some artificial cell doublets that can happen where you fix two cells together. And that is really best distinguished by sorting out cells that have a 1N or 2N DNA content. And so for this analysis, we essentially entirely focus on G1 phase of the cell cycle. But it's an interesting question. I actually don't know of any data set out there that has actually looked at different cell types in different stages of the cell cycle, like two different lineages in S phase or two different lineages in G2. There are studies out there looking at mitosis. The mitosis, you actually seem to lose most of these kind of chromosomal tab and compartment type features. You actually go into this very compacted state. And so I don't think that there's clear data that actually varies between cell type, but for S and G2, that would actually be a really interesting question.
Okay, the next question is this. Could you add extra modality like RNA or tag to single cell high-C sequencing platform to better identify cell types and connect expression or accessibility to chromatin structures?
Yeah, so you can absolutely do this. Our version of this co-assay of looking at DNA methylation and 3D genome organization was really kind of the first version of these co-assays that came online for looking at 3D genome structure. But since then, there are versions that have been published now that look at DNA, 3D genome organization plus RNA or 3D genome organization plus attack, exactly what you're seeing. And so I think that it is really nice to be able to have those kind of additional features from the standpoint of cell type identification. I think in particular for RNA, I mean, RNA is going to be extraordinarily strong in terms of identifying the particular cell type in question. As of yet, I don't think that there's particularly good methods that would look at DNA methylation, 3D genome organization, and RNA simultaneously. And so some of these cases where we are seeing these kind of, differences between the two modalities, I don't really think that we actually have a great handle of what that is related to in terms of an expression state. But we're working on ways of trying to add additional information to these assays to look at things like RNA or other sort of like histone modification states and whatnot, which I think is really exciting, but that's still very preliminary work.
Okay. The next question is, what would be the advantages of single-cell Hi-C other than the ability to identify various cell types or status? Would the resolution or quality good enough to identify differentiation pathway or trajectories, etc.?
Yeah, I mean, personally for me, I really like 3D genome organization, so I'm of course going to be interested in it. I do think that one of the clear values in my mind is that we know that these changes in 3D genome structure between cells are related to cell type specific patterns of gene expression. But not only that, they're related to linking distal regulatory elements to the target genes that they may regulate. And so I think that one of the really interesting long-term values of this is actually in terms of understanding things like genetic variation that's associated with human disease. So in cases where you may be able to find a SNP that may be affecting a distal enhancer, this can actually help narrow down sort of what is the potential target gene. And so if there's a way of getting at sort of pathway type information, I really think that would be the way that would be the most promising to use the data in the future.
Okay, this is another question from Dr. Cholgi Park, the Department of Neurosurgery in our hospital. Have you any chance to look into the 3D genome structure changes along with individual chromosomal telomere length changes?
So we have not been able to look at that. It's a very interesting question. Using short read data, resolving telomere length will be challenging. It's possible that you could actually do that. There are variants of Hi-C that you can actually do now with like nanopore sequencing, where you digest the genome, you concatenate these interactions together, and then you can sequence across the whole concatemer, and so you can get both long read information as well as what different regions of the genome are interacting with. I think you'd have to use a method like that to really begin to understand more about telomere length and how it may be contributing or related to spatial positioning of chromosomes. But I don't know if that data exists. There's only a few papers out there that have used these kind of long read type Hi-C related methods.
Yeah, I think the telomere length may be different between cell types or different individuals. So that's a related question. So my question is that have you ever compared the chromatin structure between different individuals? They may have different sequence or mutations or something like that. So is there any difference?
Yeah, so there are definitely studies out there comparing between individuals. The differences are fairly uncommon. I mean, it's a pretty small minority of the genome that we can see differences between cell types, or difference between individuals within the same cell type. I think one of the major reasons for that is, well, I guess there's kind of two. If you look at like historical, population level sequence variation pattern, it's fairly clear that the regions of the genome that are associated with these things like boundaries between domains or the anchors of chromatin loops, they're clearly under sort of negative selection. So there's a global lack of, say, structural variants or individual polymorphisms that are actually affecting these kind of regions. And so I think that they are being selected against in evolution. So I think that that's one thing that contributes to a relative homogeneity that you see between individuals. But there are some sites. There's actually really nice data out there where people have done trio studies where you have like mom, dad, and the kid, and you can actually begin to see, like there's maybe a Mendelian association. But again, they're relatively small portions of the genome that are involved in those kind of differences.
Okay, another question. Thank you for your insightful lecture. From your perspective, what is the next frontier in 3D genomics in the area of cancer research? How do you plan to continue or further develop your research?
Yeah, so that's a great question. I mean, so I think that I didn't really show a lot of this, but we've also been involved with work where if we're looking at structural variants, you can see that cancer genomes are rampant with structural variants. You have hundreds of rearrangements in any individual cell. The issue is if you then correlate this with changes in gene expression, most of those structural variants are not associated or causing any kind of change in gene expression, but there's a really small portion that are having huge impacts. And those huge impacts are, in many cases, actually potentially causal mutations, right? So these are things like immunoglobulin rearrangements that are happening in the context of lymphomas. And so those are really hard things to study because they're non-coding mutations. We don't necessarily know the rules of what kind of compatibility occurs when these kind of rearrangements take place. And so I actually think that that is a really kind of open area in terms of trying to understand, could you actually use, could you build predictive models of the impact of structural variants on nearby gene expression, which I think is really going to have to take into account aspects of genome organization. Because I think that in the long term, that could be really powerful. We don't actually sequence a lot of the genome of individual patients if you're doing things like, panel sequencing. And so there's actually a lot of genetic information in these tumors that we're actually not really using. And I think a major reason for that is we don't actually have the tools to interpret it properly. And so that's one thing that I'm particularly excited about, and I think will actually be kind of interesting and impactful in the future.
Okay, I think the Dong-Seung Lee now he's a professor in our university. Works very actively with the very similar topics with you. So I think because that there is many questions about this field. So I think someday you can visit Korea and make discussion or put some collaboration in person with many people. So thank you very much for your lecture. And,
Fantastic, thank you.
Yeah, have a good night. Thank you.
Thank you, you too.